Okay, so the process of translation, which I just modeled for you, is slightly different in prokaryotes and eukaryotes again, so there are some differences. First step is initiation. You have a start codon AUG, and it codes for the amino acid methionine. The ribosome does recognize AUG, and so it will bind to that AUG start site. Now, in prokaryotic initiation, not only does it recognize AUG, but it has to have something called an RBS, ribosome bind-in sequence, in order for the ribosome to actually latch onto it. So, just so you know, in prokaryotic initiation, you need RBS. So RBS is just the ribosome bind-in sites. Here is my RBS. So here's the sequence that the ribosome recognizes. In prokaryotes, it latches on, and then it will read the start codon and then start translation. Another difference is the initiator tRNA. I just said that AUG is the start codon, which is true, but in prokaryotes, it doesn't code for methionine. It codes for nitrophenol methionine, or FMET for short. So that is the difference. In prokaryotes, FMET. In eukaryotes, it's MET, or methionine. Another difference, large ribosomal subunit binds forming EPA sites. So how the ribosome comes together on the mRNA is slightly different. But then after that, very similar processes. So here's a diagram showing how it happens. The small subunit recognizes the RBS, then the mRNA will come in, and then you get FMET first, and then the large ribosomal will come in. So it's just a different setup than what I just showed you with eukaryotic. But I'm pretty sure all you really have to know is FMET and RBS. Eukaryotic initiation, similar but way more complex. Notice all these funny little colorful shapes here. That's the initiation complex. You have to form that before the ribosome can bind to it. We have methionine, or MET, so that's the difference between prokaryotics and eukaryotics, and it doesn't have RBS. Instead, the ribosome recognizes the GCAP, which we talked about yesterday. mRNA gets modified. You have a GCAP, a 5' GCAP. What goes on the end of an mRNA? 3 GCAP. It's not a GCAP. It's a poly-A tail. What's the purpose of the poly-A tail?
So it can get like cut off and stuff, and it won't mess up the rest of them.
It extends the life of the mRNA. Because as soon as the
That's
mRNA
how it does,
enters the cytoplasm, it's going to be degraded.
Yeah.
So it's kind of like bait, or I don't know.
So you was sort of on the right track. Like 10%.
I mean, that's kind of what I meant.
All right, and then elongation, which I just modeled for you, but you have your incumbent amino acyl tRNA, and there's the A site. Ribosome moves over, peptide bond forms between the P site and the A site amino acids. The translocation just means the ribosome moves down, process continues. Ribosome moves in a 5 to 3 direction. tRNAs, as you saw, move in the opposite. So you get the ribosome moving this way, and the tRNAs moving this way.
So the tRNA moves 3 to 5 then?
Yes. Yep. There is a little bit of energy involved in this, and this energy is GTP, but you don't need to know values.
What does that stand for? Probably triphosphate.
It's
Glycerol.
It's not glycerol. No. It's like guanine triphosphate, because they're all like based off of nucleotides. So diagram. I've already walked through this. It's just another diagram. But this is where it shows the energy. So in order for the ribosome to translocate or move over, it requires a little bit of energy. Okay, self-quiz. I know I've said this before, but how many amino acids are there?
4. 20.
20.
Why do I always say 4? Because that's the nucleotides.
No. How many mRNA codons code for for amino acids?
3.
Is it the 64?
No.
Oh, it's 61.
It's 61.
Oh yeah,
I always say
totally
3.
worked.
So how many start codons are there?
One.
One.
AUG.
AUG. So yeah, that's the start codon AUG. How many stop codons are there?
3.
3. Okay. So we mentioned earlier that there's different amino acid synthase, the things that make the tRNAs latch onto amino acids. So could we expect 61 different tRNAs with 61 different anticodons? That's what we should expect. But in reality, we've only found that there are 40 to 45 different tRNAs. And so now the question is, how does that work? Well, I don't know if you've noticed it on that assignment, on your first assignment Friday, how when you were trying to figure out the amino acid sequence, you noticed that the GA, like if you had GA or GG, if it matched the first two, the last one really didn't matter. Now, let me move on. Sorry, right here. So you notice here we have like UU, UU, and both of these code for phenylalanine. UUA, UUG, which are both purines coded for leucine. This one, if it was CC, no matter what, it was going to be proline. So maybe you noticed that for your assignments. Well, that all has everything to do with the Wobble hypothesis. So many amino acids specified are specified by more than one codon. What did we call that term? Where there are more codons for one amino acid. Starts with the letter R. Redundant. Your book might even say degenerate, but I think redundant is the word that's being used. So many amino acids are specified for more than one codon, and that, codons for the same amino acid tend to have the same first and second nucleotides, like CC or UU. So like CU, C, and CUU both code for glutamine. The anticodon GAG can base pair with both in mRNA. So as long as it matches the first two, the third one may not matter at all. So this is why it's called the Wobble hypothesis, because the first two match perfectly with the mRNA, but the last set may not. So like notice that G and C normally pair together for leucine, but leucine can also pair with C, U, U. So the first two are correct, but G doesn't pair with U. What should pair with U? A. But because we have the first two match correctly, the third one may not matter. Like it's not as important. So that's why they call it the Wobble hypothesis, because it's like six.
Wobble.
Yeah. Oops. Why did I go back? So Crick, who worked with Watson, proposed that in ribosomes, tRNAs with the non-standard third base pair can still bind successfully to that anticodon. So it's limited flexibility because 2 out of the 3 have to be correct. And so that's why you call it the Wobble hypothesis. So the non-standard base pair in the third, well, it could be CU or C or U for this G. So that's the picture showing, again, GAU, GAC, aspartame. So now the next question is, how does protein synthesis end? So we're chugging along, reading our codons that are on the mRNA, and remember there are 3 stop codons. Okay, so in this example they give you UGA for a stop codon. And the reason why there are 3 stop codons is because for those 3 codons, there isn't a tRNA. There's something that can bind to it, but it can't carry an amino acid. So then when the ribosome moves over, a peptide bond doesn't form between the last amino acid and no amino acid. And everything just kind of falls away. So other stop codons, UAA, UAG, UGA. So there are no amino acid tRNAs for those 3 codons, and that's why they're considered stop codons. So once a stop codon gets recognized and it moves over, the last tRNA detaches from that amino acid. This polypeptide chain will fold into a protein, hopefully, and then everything just dissociates or falls away, falls apart, and these ribosomal subunits will go find another initiation complex and redo its job. Ribosomes can be found in two places. You can have free ribosomes that are in the cytosol or the cytoplasm, or you can have ribosomes that are bound to the ER. When it's bound to the ER, what is the ER called? Rough ER. Okay. Those that are in the cytoplasm, they basically just make proteins, and the proteins work in the cytoplasm. Those that are found on the ER usually send those proteins to the Golgi apparatus, where they might become lysosomes, vacuoles, part of the cell membrane, and even leave outside the cell like insulin does. Ribosomes do have the ability to flip-flop their locations. They can be in the cytoplasm, and then they can send out a signal saying, hey, I want to be on the ER, and they can just go back and forth. So process of the synthesis begins in the cytoplasm. It will continue this process to completion unless it has a signal, in this case, a signal sequence recognition particle, or SRP, which will then it will migrate to the ER. So the SRP is another adapter molecule between the ribosome and the ER, and it helps it dock the ribosome to the ER. So I have a diagram that shows this. Here's my ribosome. It's already latched onto the mRNA. Okay? It has a signal sequence that says, hey, I need to go to the ER because this protein is going to become the cell membrane or something. So here's my signal sequence. An SRP will latch onto that signal sequence and bring it over to the ER where it docks. And then what happens with this signal sequence is that it starts to like go into this little channel that can get into the ER, and then your protein starts to get translated. Once it's done, the protein falls away, everything falls apart, and this protein gets folded, packaged into a vesicle, and goes where it needs to go. So the SRP is this adapter molecule that recognizes a signal sequence, brings the ribosome over to the ER.
Now it's only prokaryotes, right?
No, do prokaryotes have ER?
I thought you said the SRP only happens.
No, that's SRB, or the S.
RBS.
RBS, thank you, RBS.
Oh.
That's the RBS.
And this is the SRP.
Yeah. Okay, so that does it for today's lecture. Some questions at the end. 1, 2, 3, multiple choice. Actually, most of them are multiple choice. 6 is talking about the Wobble Hypothesis. 7, you actually have to draw a picture of a tRNA and what it does. Some more short answers.